Hi, my name is Mike Zyskowski. I'm a Senior Program Manager in Microsoft Research, and today I'm introducing the Biology extension for Excel, an add-in for Microsoft Office Excel 2007 and 2010 that exposes the features of the Microsoft Biology Foundation. Biologists and researchers will find this tool to be helpful in performing their research and making new discoveries in the field of genomics. In this video, we'll show some of the new features that are added to the Excel interface, which allow for importing a variety of biological data into Excel, comparing DNA strings from multiple sources, accessing BLAST web services, and viewing the results of the query in multiple formats. In this video, we'll take a look at genomic interval operations and the creation of a Venn diagram as a result. Here you can see we've got a worksheet open in Excel with a Bioinformatics tab at the top that's provided as part of the BioExcel add-in. There's a series of import and export capabilities, alignment functions, assembly functions, BLAST queries, and then even some built-in charting capabilities. What I'd like to focus on here, though, is the capability to operate on genomic intervals inside Excel. These are functions and capabilities similar to what you might find in some web service operations in the community today, but we've enabled the native functionality inside Excel to make it a little bit easier to work with data in its native format. One of the interesting things to do is a merge or an intersect of two different species. Here I've got a bonobo, here I've got a gorilla, and then here I have the human reference. And the human actually has metadata associated with it. Different gene information has been identified on chromosome 1 in these start and stop regions. The bonobo and the gorilla are relatively undefined at this point. And so the analysis would be to do a three-way comparison between the bonobo, the gorilla, and the human. One of the things we've done is taken the individual capabilities that are exposed here and wrapped them into a script that will actually generate a Venn diagram using 2 or 3 different inputs for similarity. So what I'll do here is I'm going to go ahead and select the bonobo data, and because I imported it using the bed format, I've got a pre-selected named region of the data, which makes it easy to select that data. Range B will then be the gorilla tab, and I'll do the same thing here. And then I'll add a sequence, and I'll go find my human reference. And my human reference, auspices of time, I'm going to select a good chunk of this data, but I'm not going to select it all. And I'll hit this, and then I'll hit OK. And now the script and the functionality provided by the Microsoft Biology Foundation is in the background, churning on this data, doing sorts, merges, and intersects between these 3 species. And now you can see that the NodeXL program, also an open source project provided by Microsoft, is being used to generate a Venn diagram that shows the overlap between the bonobo, the gorilla, and the human, and the similarity between each of them, and then all 3 of them in the middle. And the information in the data, and the regions, and their stop and starts, and the similarity thereof can all be seen in the left-hand side window, and can be drilled into further by the researcher. The value of the Venn diagram is then the ability to help disseminate this information in papers or in other means for visual clarity around the results of the comparison. Thanks for taking the time to check out this video. To learn more about the Microsoft Biology Foundation, including more videos and download information, be sure to check out research.microsoft.com/bio. Thanks.